Okay, hello. We're going to talk about the evolution of seed plants. So, seed plants show up in the fossil record about 360 million years ago, and they rapidly became the dominant plants on Earth because the adaptation of the seed was just so revolutionary and so advantageous. So, what is a seed? A seed is an embryo, so the baby plant, plus its own little food supply and protective coat, all in one. So this is a really amazingly advantageous structure because it enables offspring to survive for long periods of time away from the parent because they have this food supply and they have this protection from the environment. And they also are able to go dormant if environmental conditions are not favorable to growth, and then germinate later when conditions are favorable. So they're really, really extraordinary adaptations. I should mention the seed plants were also among the first to develop thickened woody stems, and this is also really advantageous because it allows plants to get a lot bigger and a lot taller and intercept more sunlight. So all seed plants have these 5 characteristics. Now, there are some plants we talked about earlier that have some of these. For example, there were some seedless vascular plants that are heterosporous, but all plants have these 5 adaptations. A reduced dependent gametophyte, heterospory, eggs within ovules, sperm within pollen, and secondary growth, which is growth in girth. So the trend in land plants in general is one of a continual reduction in the size of the gametophyte and an increase in the size of the sporophyte. Excuse me. So we have an evolutionary trend. Evolutionary trend away from prevalence of the gametophyte. So if we look at our different types of plants here, so here we have our seedless vascular plant. I'm sorry, that's not a seedless vascular plant, that is a non-vascular plant. So if we take something like a moss, the gametophyte is the dominant part of the alternation of generations. It's the bigger part, and the sporophyte is very small and nutritionally dependent. If we then look at the seedless vascular plants, such as ferns, the gametophyte is quite small, but nowhere, still not as small as it's going to be, but it's quite small, and the sporophyte is quite large. But the gametophyte is, in fact, nutritionally independent, so these two are actually independent of one another. We see an even more extreme reduction in gametophyte size in our seed plants. So here's an example of a flowering plant, which is a seed plant. And here, if we want to look at the gametophyte, we're not really going to be able to do that unless we get out a scalpel and a microscope and do some dissection, because the female gametophyte is actually going to be found within the sporophyte structures. So here's our female gametophyte, and they're microscopic essentially. They are, excuse me, they are multicellular in some cases, but microscopic. So very tiny, and they are retained within sporophyte tissues. So this tiny gametophyte, just like the other gametophytes in every other plant life cycle, does develop from a spore. But this spore is retained within the sporangia of the parental sporophyte. It never leaves, okay? So that's one difference. So you don't see it because it all stays enveloped within sporophyte tissue. And this is actually pretty advantageous because it protects the gametophyte from environmental stress, such as UV radiation or desiccation. and it enables the gametophyte to get nutrition from the sporophyte. Now, our gametophytes are going to be making the gametes, so they're really, really critical to the evolutionary success of the plant. So it makes sense that we want to protect them. All seed plants are also heterosporous, so they have heterospory as their life cycle. So in this life cycle of being heterosporous, we have two types of sporangia, megasporangia and microsporangia. And we might remember these. This is actually a seedless vascular plant you're looking at here. But just kind of to refresh you, we have megasporangia that produce megaspores that will germinate into a female gametophyte, and microsporangia that produce microspores that will germinate into a male gametophyte. Same thing with the seed plants, except for, again, everything is going to be retained within sporophyte tissue. So we have a megasporangium that is producing megaspores. These megaspores, in turn, will give rise to a female gametophyte. But it all happens within the sporophyte tissue, okay? That's kind of the big thing that sometimes makes people confused about seed plants. It's really the same thing we have in all of our other life cycles. It's just that everything is very small and you don't see it because it stays within the sporophyte tissue. And for the male structures, we have microsporangium that produce microspores that will give rise to the male gametophyte. Here we're looking at, this is actually, you might recognize it, it's a longitudinal section of a female cone from a pine tree or some other similar conifer. And this is a female-only structure. So in this example, you had both kinds of sporangia on one plant. That's not always the case. So here we see just the female megasporangia. Well, actually, what you're looking at is many megasporangia. So each of these is a megasporangia. And let's look at that a little closer to examine ovules and eggs. Ovules and egg production. Okay, so remember everything here is going to be retained within the sporophyte. So let's talk about this megasporangium. So here's our pollen cone. Sorry, not pollen cone. Here's our female cone. And if we zoom in here, okay, there's a megasporangium. So what does this thing look like? So we've got this fleshy megasporangium, which is protected by a layer called the integument. This integument actually is later going to become the protective seed coat of the seed. I'm sorry for the spoiler. So within this megasporangium, we have this megaspore here. This structure right here. The megaspore is going to develop into the female gametophyte, which produces an egg. Actually, I want to use the word megaspore. So what you're looking at here in this figure is progression of stages of development. So here we have the megaspore here, and then in this next figure, this megaspore has developed into a multicellular, still very tiny, but multicellular haploid female gametophyte, which has produced an egg. Now in this figure, we also have some of the male stuff, the pollen grain meeting it. Just ignore this for now. I'm trying to cross it. Ignore this for now. We just want to focus on female structures for now. We'll talk about the male structures in a minute. Okay, but I just want to make this clear because this gets people confused. Because again, everything stays surrounded within sporophyte tissue. So it's really the same life cycle that we saw in the mosses or the ferns, just the gametophyte's really little. So again, we have our megasporangium, and in this case, it's surrounded by that integument. Within this megasporangium, we find a megaspore that will develop into a female gametophyte that produces an egg. And the whole structure, and by which I mean the megasporangium, the megaspore within it, plus the integument, is collectively called an ovule. So usually when we talk about seed plants, we talk about ovules producing eggs. And that is correct, but we need to understand that an ovule includes the megasporangium, the megaspore, and the integument. So there's a lot going on there within that structure we call an ovule. All right, let's talk about pollen and sperm production. So again, this is a heterosporous life cycle. So if megaspores make eggs, microspores are going to make the sperm. So we have a microsporangium, which is right here. Which contains microspores. These microspores are going to develop into a pollen grain. Which consists of the male gametophyte enclosed within a protective pollen wall. So basically it's just the male gametophyte packaged into a protective coating. So let's take a look at this. So we have our microsporangium, which we sometimes call a pollen sac. And in here we have specialized cells that will become the microspores. Each of these microspores is going to become part of a pollen grain. So each pollen grain actually has two nuclei surrounded by a coat. So this is essentially the male gametophyte surrounded by a protective coat. And the coating of the pollen is actually one of the most strong and environmentally resistant substances that we know of. It's a really fabulous adaptation to resist drying and things like UV radiation. And as you know, if you have allergies or if you've parked under a tree, these plants, seed plants, produce Millions of pollen grains. They're everywhere. So the whole point here is that we want pollen to make its way to the ovule and to undergo the process of pollination. Transfer of pollen to the area where the ovules are. And so a really cool thing's going to happen when the pollen grain gets within proximity of the ovule. It is going to germinate and when it germinates, it will generate a pollen tube So let's look at this structure again, but think about the males, the male part here. So this is a cool thing. So here's a pollen grain, and when it gets in proximity of the female, and usually there's going to be some kind of moisture that will help enable this pollen and signal, and also chemical signals that signal to this pollen grain to germinate. Because as I said, that pollen wall is very tough and strong and resistant. So it germinates a pollen tube. One of the nuclei here is actually called the tube cell nucleus because it's going to germinate and elongate into that pollen tube. And the other sperm here is called the generative, or I should say the other nucleus here is called the generative nucleus. It's going to split and form sperm. So we grow a pollen tube and sperm gets sent down this pollen tube and discharged into the egg of the female gametophyte. And when we have the union of the sperm and egg, that is, of course, fertilization, yay, which is really the goal. So pollination is just getting the pollen to the ovules. Fertilization is when we actually have the union of egg and sperm, which is the real goal for reproduction. And then if we look at our next stage of development, what happens? Well, the seed coat is going to start to toughen up. The integument is going to start to toughen up and become a seed coat. And we are going to have a new baby sporophyte developing within that tissue of the female gametophyte, which in the case of the conifers and a few other plants, this will be the food supply. Okay, so finally I want to talk about the last point of secondary growth. So seed plants are the first to possess this type of growth, and it's all thanks to a tissue called a lateral meristem. These lateral meristems, one is called the vascular cambium, which gives rise to what we call wood, and one is called the cork cambium, which gives rise to what we call bark. And in chapter 33, we're going to discuss this in more detail. But the main result of these two types of tissues is that we get, growth in girth. So we have this is our vascular cambium, which is giving us growth in girth by adding layers of wood. And we have our cork cambium, which is right here, that is giving us a nice thick bark as a really, really nice protective layer against the elements. So both of these two things are really, really advantageous because they give you great protection against things like, in addition to environmental challenges like desiccation, bark is a great protection against pathogens or pests. And also, having wood being very strong allows you to get very tall without falling over. So it also allowed seed plants to get really, really, really big and tall. Okay, I'm going to pause here. We'll pick up in the next video talking about a little bit about our different seed plant groups. So see you then.